This practical is designed to investigate some sites of drug action using the isolated ileum. To that end, you will be divided into groups, and each group will have a piece of apparatus consisting of an organ bath and a computer. If you look at the organ bath, you will find that the organ bath is sitting in front of you, and inside that organ bath you have a glass tube, like so, and outside of that you have liquid, which in this case is just water, and inside here you have an additional liquid and a piece of tissue, which is anchored at the bottom with a piece of string coming off it. The water bath on the outside is at 32 degrees C. So please check your organ bath at the front with the dial and make sure that it's set to 32. Why do we set it to 32? This is a question that you need to think about. If you think about it for a second, and then see if you can come up with an answer. The body temperature of a guinea pig is 37. And as we have been talking about in the autonomic lectures, we were talking about the motility of the gastrointestinal tract, and we were talking about peristalsis. So what do you think happens at 37 degrees C? Yes, that's right, peristalsis. If you have a tissue that is already contracting, how would you be able to determine the effect of the drug that you are acting? The answer is you would not be able to. So by reducing it to 32 degrees C, what we actually do is we reduce the chances of peristalsis. That does not mean that your tissue will not become spontaneously active. This does happen, as we talked about. The enteric nervous system is present in the ileum, and as a result will actually cause motility, will cause the gut to contract. But hopefully at 32 degrees C, that we maintain it at a lower level, and as a result, that when you actually add your drugs to the bath, you will actually see the effect of a drug and not effect of the enteric nervous system. What about the tissue itself? The tissue is sitting here inside a liquid. Do you think this liquid is water? No, it isn't. If you put your tissue into water, nothing would happen. So that liquid in with the tissue is something called a buffer. In this case, something called tie roads. What do you think is actually in that buffer? Well, everything that the tissue needs to be able to work. So it contains all the salts, glucose, sodium, potassium, all of the material that it requires to be able to contract. So it can provide energy, and it has obviously the ions enabling it to be able to depolarize the cells and contract. In addition to that, what else does a tissue actually require? If a tissue is contracting, it requires energy. And to get energy, what do you actually need? You need your tissue, your cells, to actually make ATP. And we make ATP using oxygen, which if you look at the actual bath, you should see bubbles running up it. That is because we are bubbling the buffer with air. Now, do you think that tissue is actually happy being bubbled with air? If you think about it for a second. No, it isn't. When you are bubbling it with air, you have got 20% oxygen that you are trying to dissolve into that buffer to give enough oxygen to that tissue. Do you actually think that we can actually dissolve enough oxygen in a liquid, in water, to actually keep the tissue happy? If you think about it again, the answer would be no, you can't. If you could, the body would not bother going to the process of creating red blood cells. We create red blood cells to carry oxygen. And without the red blood cells, we could not dissolve enough oxygen inside our blood to get enough to our tissues, and as a result of that, we would not be able to produce as much energy as we require. So, this tissue is under hypoxic conditions, which means it's not totally happy, but it will survive for the length of the experiment, in this case, about 4 hours. So that's the setup so far. What's the piece of string for? The piece of string runs up to what should look like a little black box. And that little black box detects contractions, detects movement of that tissue. When that tissue contracts, it pulls on the muscle. It pulls on the string as well. And by pulling on the string, it pulls down on the black box, and that sends a signal to the computer. It is a transducer. And it turns mechanical movement, that is the contraction of the tissue, into electrical signals which the computer can pick up and register on your screens. It is set up so that the actual tissue has approximately 1 gram of tension on it. Why would we already stretch the tissue by putting 1 gram of tension on it? Well, if you think about it, if the tissue was just sitting there with no tension on it, it would just be flapping around in the bubbles in the actual buffer. And when it contracted, it would pull on the string, perhaps, but not very hard. By putting 1 gram of tension on it means that actually when it pulls against the tissue, when it contracts, it is actually pulling very hard. So you can actually detect the signal. So it needs this bit of tension so that we can actually be able to see the contraction when it happens. So that's the actual setup. It's all fairly straightforward. So the next part is to move on to the actual experiment itself and the parts that you will be carrying out today. Okay, the experiment. We've broken the experiments up into various parts. And the first one you're going to do is part 1. So what part 1 involves is you adding ACH to the bath to get 1 times 10 to the minus 7 molar inside the actual bath. That's the concentration that you want in the bath. Why are we doing this part 1? Mainly because this tissue was in the guinea pig not so long ago. We've taken it out, we've reduced it to 32 degrees. So what we actually need to do is we need to be able to get a response out of the guinea pig that is consistent. You need to almost warm the tissue back up. So what part 1 involves is adding 1 times 10 to the minus 7 molar ACH in a 2-minute dose cycle. So that is that at time 0 you will add the drug. You will wait 20 seconds and you have a timer to work that out before washing out. After 60 seconds you will wash out again, and then at 120 seconds you start the whole process again. And what you are looking for is you are looking for 3 peaks which are approximately the same size that you can see on your computer screen. And what you might find is that the first peak that you have is quite small, then the second one is a bit bigger, and then the third one, and there you go. And there you have your 3 peaks. Approximately the same size. They don't have to be exact, but as long as they're close, then in which case you can proceed on to part 2. So the question is how do you actually get 1 times 10 to the minus 7 molar inside your actual bath? Well, here's the mathematics involved, and this involves some calculations. The bath that you have with your piece of tissue in it is 25 mils. That is equivalent to 25,000 microliters. When you add drug to that bath, what we are going to ask you to add is 0.25 of a mil, or 250 microliters. So if we're adding 250 microliters to a 25 mil bath, 250 microliters into 25,000 microliters, what is the dilution factor? Take your time, think about it, come up with an answer. Okay, hopefully you've recognized that that is a 1 in 100 dilution. So when you add 250 microliters into a 25 mil bath, 25,000 microliters, that is a 1 in 100 dilution. I've told you that you need 1 times 10 to the minus 7 inside the bath. So, in which case, in the test tube, what concentration of ACH do you need? 0.25 mils going to 25 mils, 1 in 100 dilution. What concentration of ACH do you need in the test tube? Think about it for a second, pause the video if you need to, and work through the calculations. Okay, you should have worked out that you need 1 times 10 to the minus 5 molar. If you haven't, stop the video again and have a look and see if you can work it out. But essentially, 1 times 10 to the 5 molar is 100 times more concentrated than 1 times 10 to the minus 7 molar, which means that when you take 250 microliters of it and put it inside a 225,000 microliter bath, you're diluting it 1 in 100, so as a result it's going from 1 times 10 to the minus 5 to 1 times 10 to the minus 7 molar. You need to understand the dilution factors here. This practical is based all around these sort of basic mathematics. If you get this bit right, then the practical will work and there will not be a problem. Okay, so what you need for part 1 is that you need 1 times 10 to the minus 5 molar ACH. And what do we actually give you? Your stock is 1 millimolar. 1 millimolar is 1 times 10 to the minus 3 molar. So you need to go from 1 times 10 to the minus 3 molar to 1 times 10 to the minus 5 molar for you to complete part 1. So how are you going to do that? First of all, how much do you have to dilute it by? 1 times 10 to the minus 3 molar, and you want 1 times 10 to the minus 5. So that is a 1 in what dilution? Think about it, work it out. Stop the video if you need to get a piece of paper to work it out. Yes, it's a 1 in 100 dilution. So from the stock solution that we've given you, 1 times 10 to the minus 3 molar, 1 millimolar, you need to dilute that 1 in 100 before you start part 1. So how are you going to do that? Well, 1 in 100. What is 1 in 100? In theory, you could take 1 mil of your stock, you could add 99 mils, and in this case, it's of the tie Rhodes buffer. You will be doing all your dilutions in the tie Rhodes buffer. So you could take 1 mil and add 99 mils, and that would be 1 in 100. But that is incredibly wasteful when you consider that you only need 250 microliters at a time of it. So we would rather you didn't make up 100 mils of the stocks. So how are you going to do it instead? Well, what you can do is that you can make 1 times 10 to the minus 4 molar first. 1 times 10 to the minus 3 to 1 times 10 to the minus 4 is what dilution? It's a 1 in 10. And so how do you get the 1 in 10 dilution? Well, that's a bit more realistic. It's going to be 1 mil of your stock at 1 times 10 to the minus 3 plus 9 mils of your buffer. And that will give you 1 times 10 to the minus 4. How do you go from 1 times 10 to the minus 4 to 1 times 10 to the minus 5? Well, yes, it's a 1 in 10 dilution again. So you take 1 mil of your 1 times 10 to the minus 4 and you add it to 9 mils of buffer, and there you go. You have your 1 times 10 to the minus 5. You will probably already look at the solutions and you will see that they are all colorless. So label your tubes. Label them with the substance, ACH, or whatever you might be diluting, and label it with the concentration that's inside the tube. So you should have 3 tubes before you start part 1, labeled ACH, 1 times 10 to the minus 3 molar, ACH, 1 times 10 to the minus 4 molar, and ACH, 1 times 10 to the minus 5 molar. Once you have the 1 times 10 to the minus 5 molar, then you can start part 1. But don't throw away 1 times 10 to the minus 4. You will need it for later. Okay, that's part 1. Now you can move on to part 2. So what we're looking for part 2 is a dose response curve to ACH. And we want you to do a dose response curve starting at 1 times 10 to the minus 8 molar, 3 times 10 to the minus 8 molar, 1 times 10 to the minus 7 molar, 3 times 10 to the minus 7 molar, 1 times 10 to the minus 6 molar, and finally 3 times 10 to the minus 6 molar. And these are all the concentrations inside the bath. So you've got those concentrations inside the bath that you need. What are going to be the concentrations that you need inside the actual test tubes? Well, we've already gone over the first bit, which is you wanted 1 times 10 to the minus 7 for part 1. And as we worked out that you need 1 times 10 to the 5 molar in the test tube for 1 times 10 to the minus 7. Because you're adding, remember, 0.25 mils into 25 mils, and that's 1 in 100 dilution. So, if you need 1 times 10 to the minus 5 to get 1 times 10 to the minus 7, what do you need for 1 times 10 to the minus 6? Well, you need 100 times concentrated, which is 1 times 10 to the minus what? Have a think about it. Should be fairly straightforward. Yeah. It's 1 times 10 to the minus 4 molar. Something that you've already just made up, which is why you keep the tube, and which is also why you label the tube, because you're going to need it for part 2. So following on from that, it's all very straightforward that you need 1 times 10 to the minus 6 molar inside the test tube to get your 1 times 10 to the minus 8. And how do you get these? Well, we told you that the stock was 1 times 10 to the minus 3 molar. So you did a 1 in 10, 1 in 10, 1 in 10. That's what you'll do first of all, remembering that you're diluting in Tyrode's buffer. Okay, well, that seems fairly straightforward. What about 3 times 10 to the minus 6? What do you need inside the test tube to get that? We'll need 100 times concentrated, which is going to be 3 times 10 to the minus what? Yep, that's right. It's 10 to the minus 3 times 10 to the minus 4 molar. Ooh, how are you going to get that? Well, it's fairly straightforward. You took 1 mil of the 1 times 10 to the minus 3, added 9 mils of Tyrodes to get 1 times 10 to the minus 4. What are you going to take to get 3 times 10 to the minus 4? Well, it's 3 times more concentrated. So instead of taking 1 mil, you'll take 3 mils. And then you'll add 7 mils of buffer to it. And that will get you your 3 times 10 to the minus 4. Okay, working on that basis, you need 3 times 10 to the minus 5 molar, and you need 3 times 10 to the minus 6 molar. You may have seen that there are 2 ways of being able to do this. Once you've got your 3 times 10 to the minus 4, you can do the usual 1 in 10 and the usual 1 in 10 to go down the concentrations. 3 times 10 to the minus 4, a 1 in 10 dilution will become 3 times 10 to the minus 5, and a 1 in 10 dilution of 3 times 10 to the minus 5 will come 3 times 10 to the minus 6. So you can do it that way. Alternatively, you can do it the way you did it to start off with, from the 1 times 10 to the minus 3, taking 3 mils and get 7 mils to get 3 times 10 to the minus 4. Well, you can take 3 mils of 1 times 10 to the minus 4, add 7 mils of buffer, and you'll get 3 times 10 to the minus 5. And just the same, you can take 3 mils of the 1 times 10 to the minus 5, add 7 mils of buffer, and get 3 times 10 to the minus 6. Whichever way you do it is fine with me. Once you have all those concentrations inside their tubes, all well labeled, then you can start part 2. And what we're looking for in part 2 is one single peak for all these concentrations. And you use the 2-minute dose cycle. So what we're expecting you to do is at time 0, add the drug, 1 times 10 to the minus 6 molar to the bath to get 1 times 10 to the minus 8. Do your washout at 20 seconds, your washout at 60 seconds, and then at 120 seconds, you add the next one. And then you add the next one 2 minutes after that, 2 minutes after that, 2 minutes after that, 2 minutes after that. So that whole part should take, you got 6 concentrations, 2 minutes, 12 minutes, and part 2 should be done. I suggest you work up the concentrations, not down, and I suggest you do it in the order that we suggest in the actual practical schedule. Okay, You've done parts 1, you've done parts 2, you have your dose response curve, which you're going to need for the actual write-up. Now we're going to move on to part 3. So what we're going to do with part 3 is that you are going to add and test the effect of morphine on the ACH response. So how are you going to do that? First of all, you're going to select the submaximal dose of ACH, something that gives you a response but not the maximum response. And usually people have found that to be 1 times 10 to the minus 7 molar ACH, exactly the same as what's in part 1. And what you're going to do first of all is that you are going to get a 1 times 10 to the minus 7 response using the usual 2-minute cycle. That's your control response. You need a control response. If you don't have a control response, how are you going to know that morphine has had any effect whatsoever? Then you're going to suggest and start with a concentration of morphine. Again, with morphine, we have given you it at 1 times 10 to the minus 3 molar, i.e. 1 millimolar. And what you should do is you should go back to the data that you received and that you got from Dr. Allen and work out what concentration of morphine had the maximal effect, and that's the concentration that you're going to try. Essentially, the best concentration to try is 1 micromolar. What's 1 micromolar? 1 times 10 to the minus 6 molar. So you want 1 times 10 to the minus 6 molar of morphine inside the bath. So similarly, as you did with ACH, you need to make a dilution from your stock, which is 1 times 10 to the minus 3, down to 1 times 10 to the minus 4 molar. So it's a 1 in 10 dilution. And that's the concentration of morphine you're going to use. Then if you look at part 3, what you're actually going to do is that then you are going to do a 3-minute dose cycle. So at one point, the very start, you're going to add your morphine. You're going to wait 60 seconds. Then you're going to add your ACH. And you're going to see what happens. You are then going to wash it out at 80 seconds and at 120 seconds. And then at 180 seconds, you are going to start all over again. Except that you're actually going to do is part 4. You are going to see what the effect of morphine is, and you just need to do it once. So it'll take you 3 minutes to do that part. Then you're going to move on to part 4. And part 4 is exactly the same as part 3, except that what you're trying is naloxone. So what you're going to do is you're going to have a little look at, back at your data that Dr. Allen gave you, and you're going to have a look and see which concentration of naloxone reversed the effect of morphine. Again, we'll recommend that you do 1 times 10 to the minus 6 molar of naloxone. And again, it's coming to you in the stock at 1 times 10 to the minus 3 molar. You need to dilute that 1 in 10 to get 1 to 10 minus 4 molar, and that's what you add to the bath to get 1 times 10 to the minus 6. So again, what you're going to do after this part, for part 3, is again, you're going to do a 2-minute cycle to get your ACH control. You need the control again. As time goes on, the tissue becomes more worse for wear. So the responses might go down a little bit, so you constantly need to keep redoing that control. After you've done the control response, you're going to do the 3-minute cycle again, except this time you're going to add the naloxone. You're going to wait a minute, then you're going to add the ACH, and then you're going to see what the effect is. At that point, you are going to do, after you've done that, another ACH response on its own in 2 minutes, just to make sure that everything is returned to normal, before moving on to part 5. In part 5, what you are going to do is that you are going to do the same as you did for parts 3 and parts 4, except this time you're going to be using atropine. You all know what atropine is, so you can all predict what effect you're going to see. Atropine is slightly different to morphine and to naloxone. The concentration, which we recommend you try, is 1/10 of the concentration of the ACH that you're using. So if you're using 1 times 10 to the minus 7, we would like you to use 1 times 10 to the minus 8 molar atropine. Okay, so you should all remember that atropine is an inhibitor at the muscarinic receptor. So hopefully you can all predict what the effect is when you add atropine before you add ACH. And what we're expecting is, if you've done your control response, that when you add your atropine, and then a minute later add your ACH, that you get a response. If you get exactly the same response, that does not mean that atropine doesn't work. It means you haven't used a high enough concentration. So you need to go back around and repeat the experiment again, but this time increasing the atropine concentration to the minus 1 times 10 to minus 7 molar. If, on the other hand, your response is 50% or less, in which case you will have determined that you have had an effect of atropine, and that is what we're looking for. You should always remember that atropine is competitive, so once you've got the inhibition, we want you to get the response back to ACH. So you need to go back into doing 2-minute dose cycles with ACH at 1 times 10 to the minus 7 until the response comes back. And this might take quite a few 2-minute cycles before you actually see it. Once you've got it back, that's the experiment finished. Part 6 is the data collection analysis. I'm hoping that as we have a 4-hour block that you will be able to do the data analysis inside the actual laboratory. And we will talk through the data analysis at that time.